So now we're going to start talking about our molds and our fungi, which a fungi is a mold or a yeast. Yeast are going to be single cellular and molds are going to be multicellular. And many of the species can transition between single and multicellular. We'll start first by talking about just general features of our molds and yeasts, and then we'll talk about individual classes of diseases caused by these pathogens. So mycology is the study of molds and yeast. Here is a picture of a yeast, a single cell, and then here, of course, is a mold that's infecting an individual, a chromoblastomycosis. And this is caused by a fungus that is going to be darkly pigmented, and fungi that have dark pigmentation is simply because they have melanin, similar to what our skin has, in their cell wall, so that they look darker. The yeast on the side is Cryptococcus neoformans, and this is going to be an opportunistic fungi, and this is going to cause meningoencephalitis in individuals who are immune compromised. So fungi are considered heterotrophic. They can be saprobes, which are going to survive on dead and dying material. They can also be symbiotic. So this is more in nature with respect to plants. So they can have a mutual advantage of living on an organism. They can have commensals. So this is where one benefits. And in the case of our commensal fungi, the fungi will benefit from living on us, but they don't really harm us. And so that would be a commensal interaction. They can also be parasitic, where the organism, such as the fungi, is going to benefit at the expense of us, the host. And so most, of course, of medical mycology is centered around parasitic. Now, fungi are going to be in the kingdom of fungi. They are eukaryote, and so they do have your Golgi apparatus. They do have mitochondria, so they do have organelles, so it's different than our bacteria. They also have very specific proteins in their cell walls. So they have a very thick and rigid cell wall, which is called chitin, and they have specific glucans in their cell wall also. And because these are very specific to fungi, these are some of the targets of our antifungal agents. They also in their membrane have another protein or another molecule. It's not really a protein, it's a fat, ergosterol. This is their substitute for cholesterol. And so that's going to be embedded in their cell membrane. Now, ergosterol is also going to be a target for antifungal agents as well. And again, you're going to have two kinds, multicellular, which is called our molds, and our yeast, these are single cellular. So let's just look at yeast just for a moment. Now, yeast are going to be able to divide by fission, where they simply split in half. They can also bud, so you have like a little bud building off of the original yeast cell, and those are called blastoconidia. And you can also have a tube form where you get these pseudohyphae forming. And so pseudohyphae are very important for Candida, invasive Candida species. So inside of our body, Candida is always going to be a yeast, whether it's in the environment or inside of our bodies. However, inside of our bodies, it can form these pseudohyphae, so it can look like it has long chains attached to it. And this is just some examples of our budding yeast, different mechanisms of budding. And so this is how our single cellular yeast will divide. Now, our molds, on the other hand, are going to be multicellular, and they are going to have these string-like structures that are called hyphae. Some of the hyphae you can see have these little breaks in them, and so they look almost compartmentalized. And so those are referred to as septate hyphae. They're divided. Others look like long tubes with no divisions, and those are coenocytic hyphae, hollow, multinucleated. And so your septate hyphae will have individual nuclei within each septate, whereas your coenocytic will have your multinucleated, so it will look like one giant cell with many nuclei in it. Now the hyphae are eventually going to form a molecule called a mycelium, and that's typically what we see. When we look at a mold growing somewhere, we are seeing a bunch of hyphae that are forming what is called a mycelium. Now, with the hyphae, one thing to recognize about it is that it has two different areas. You have a thick wall area that is very thick and rigid, and then you have another that is very plastic and stretches, and that's so that the hyphae can grow. And it's at this area here where you're going to have nutrients in. So I'll do that here. And you're going to have waste out. So the hyphae really at the ends are what's going to be metabolically very active with the environment, picking things up and getting rid of things. This is all going to be controlled by the nuclei in your hyphae. So the nucleus is going to send signals, and these signals are going to tell your ends to do something. Now the hyphae are going to, or your molds are going to start, for example, from a spore. So many of our fungi form spores, and when they're in the right conditions, they will germinate. And they'll germinate and start to spread their hyphae, and they'll start to branch, and eventually they'll become a mycelium. And a mycelium is basically going to be an aggregate of your branches of your hyphae. Now let's just talk a little bit about spores. So with respect to spores, we have our spores are considered asexual reproduction. You have spores that are going to be encased inside of sort of this like membrane, and this is a sporangium. And so these are called sporangispores. You're also going to have spores that are naked, and we have our naked spores are going to be called conidia. But both spores and conidia are in essence spores. They're the same thing. They just have different structural characteristics. So some examples of our sporangiospores are going to be our Mucoralis and our Rhizopus species within that. Our naked spores are going to be the sort of pretty looking flower fungi, our Aspergillus, our Penicillium, and our Dermatophytes. They are all going to have naked spores or conidia. Now you can see that we have a little bit of terminology associated with this. We have our sporangiophore, which is sort of the trunk that holds the sporangium, and encased in the sporangium is the sporangiospores. We also, on the side of the conidia, you have your conidiophore, and that is going to be the trunk for holding your conidia. And then we have, of course, our septic hyphae, which is associated with our Penicilliums, Aspergillus, Dermatophytes. And then our Rhizopus species, which has our sporangiospores, is going to have rhizoids, which are these little, they look like almost little claws that grow off of the hyphae. Now, with respect to respiration, they're all going to be aerobic. They live in the environment. They're going to interact with oxygen. They can also have some facultative, so these would be our fermenters. And you also have some that are going to be strict anaerobes, which means that they would not be exposed to oxygen. They'd be like in the ground, in the dirt, in areas where the oxygen is limited. They have primary metabolites, which is what they use, ethanol, glycerol, etc. Secondary metabolites are what they're also going to make. They make antibiotics, so they get infected by bacteria too. So a lot of our antibiotics come from our fungal friends. They also are going to make anaphyltoxins, and anaphyltoxins are going to be important for allergic responses. Now, in comparison to our bacteria pathogens, our fungal pathogens are going to grow very slow for the most part. There are a few exceptions, which we'll discuss. And so in order for them to divide, it takes time. So several hours in order to make, to bud, for example, to make a new yeast. And so they're going to grow slow in our cultures. They're also going to grow slow inside of us. And so they're going to have more insidious onset of disease. Now, of course, fungi are very well known for allergic reactions, respiratory infections. You can have different areas of the body where your fungus will grow. You can have athlete's foot, tinea pedis. You could have our sporotrichosis, which is going to be common in people who do gardening because they get pricked on their fingers with thorns and other spikes associated with plants. And those can be contaminated with our sporotrichosis, and you can get infected with that. And then there are also going to be systemic mycosis. And the systemic mycosis are going to be the mycosis that are going to be able to infect people who are immune competent. However, it's not really going to cause much of a disease in immune competent individuals. And it's really people who are at risk who are going to get disease. Coccidioides immitis is referred to as desert fever, for example. So one of the main important things to take away from this introduction is that we have fungus everywhere. We know that. Many of us associate fungi with irritation, such as allergic responses, but they are really hidden killers. And so you have to consider that anybody who is immune compromised or has some sort of disease that influences their ability of the immune response to function, those individuals are all at risk for just normal fungi in our environment. So Candida is going to be an important one we'll talk about. And Candida has a mortality rate of up to 75%. And again, these are in immune compromised individuals. And so the death rate is going to be pretty high. Cryptococcus neoformans, where you're going to present with meningitis, has up to 70% mortality. Aspergillus can have up to 95% depending on the state of the actual patient. So people who are debilitated in the hospital are exposed to Aspergillus all the time. It's known as the showerhead fungus because it grows everywhere. And we, in our immune competent states, are able to control that. We don't get affected by Aspergillus. But a person who has some sort of immune compromise, or they are debilitated, or there's something going on with them, they are highly susceptible. Pneumocystis jirovecii. This is very common for AIDS patients, and it has a very high mortality rate in those patients. We have our endemic mycoses, and we're going to talk a lot about those in their own lecture, up to 70%, and our mucormycoses, up to 90%. So again, these pathogens are not going to affect immune competent people too much. They might cause some sort of cold-like symptoms or allergic symptoms, but for the immune compromised, especially in a hospital type setting, a long care facility setting, they are really going to potentially cause a lot of problems for those individuals.